Excellent. Thank you very much for the introduction, Carl. There is a small housekeeping note. There was someone in the Q&A that said the chat box is disabled to attendees. So if we want questions and or universities, we're going to want to open that up to everyone to participate. So again, thank you very much for the invitation to present at WESTalks. It's really a great new media to be able to present some research ideas and some of the work that we've been doing to people we wouldn't otherwise be able to directly communicate with. So I really enjoy having kind of this international and across Canada focus, being able to present some of the work that we would be doing. And the presentation I'm giving today is really looking at biofiltration and where we've been, where we are now, and really what the next steps for this technology are. Looking at green technologies and biotechnologies is really a popular area of research, but also really important for future sustainability in industrial processes such as water treatment. So before I get started, I want to thank everyone who's kind of participated on this work over the last 8 or 10 years with the Drinking Water Research Group. I'm not going to name everyone, we'll be here for a while, but I just want to acknowledge some of the other co-authors who have participated and really helped to drive our success in the research that we've been doing in the area of biofiltration with our drinking water partners. So outline, we're going to talk a little bit about where we've been. So what the history and the background of biological treatment in drinking water is, an overview of some of the historical biofiltration research. I put quotes around historical. It doesn't seem like it's been that long, but as I've been doing this for almost a decade now, it seems like it's getting to be more and more diverse as we go on. Also, we'll then move to kind of a current state of the industry. Where are we now? And what has the research led to in 2020? And finally, talk about some of maybe where we're going. I think this is really probably the most interesting and exciting part of this presentation for everyone, and really the part that I hope spurs the most conversation, but talking about what are the next steps. We know what it can do, we know how to apply it, we know how to operate these systems. What can we do as researchers? What can we do as academics or people in the industry to make it better moving forward? Where we've been. So, what is biological treatment? And from a drinking water context, what we're talking about today is letting naturally occurring microorganisms consume or degrade substrate. So, this substrate is relatively loosely defined. It may be things like natural organic matter, it could be metals, it could be nutrients. There's any sort of substrate that we may want to remove, and it's typically based on what drinking water contaminants are present. So, if we're looking to remove disinfection byproduct precursors, that may be the primary substrate that we're interested in. However, other water systems may be more interested in the removal of manganese or iron. They may be required to remove nitrogen or phosphorus. So there's any number of substrates that might be able to be consumed biologically. And really that objective is what defines biological treatment at most facilities. And when we're talking about biological treatment, there's typically two types in water and wastewater. There's either suspended biomass, which you might see in an activated sludge process for wastewater, or there's fixed film. This is most common in drinking water plants, but it also is important to characterize what this is. When I talk to most people about fixed film biological treatment, this is kind of what they're envisioning. A rock covered in algae and biomass, kind of a goopy looking thing coming from the river. In reality, what we're seeing is really small accumulation of biological components on these types of systems. So looking at this SEM image at a scale of about 5 microns, we're seeing that biological accumulation may be relatively small. It's not going to necessarily be large scale biological components. Instead, it's just small accumulation on the surface of existing filter media. Looking at a brief history, starting in the early 1900s was really when drinking water at an industrial municipal scale took off. And this really stemmed from the use of chlorine gas for disinfection. This is probably the most important public health development in the last 150 years in that it saved millions and millions of people, both from getting ill and from dying in general due to waterborne illness. However, this also then became kind of this crutch that operators and water systems relied on, whereby more chlorine was better for a very long period of time. So not only were they trying to kill microorganisms and prevent illness, they were trying to prevent any sort of biological growth in their drinking water system by adding huge amounts of chlorine. In the 1930s, biological wastewater treatment really started to take off. Some of the first known literature documents were shown that biological activated sludge processes could effectively degrade organics prior to being discharged back to the environment. It was about 70 years after the introduction of chlorine gas that these chlorinated disinfection byproducts were observed. In 1974, the US EPA regulated the concentration of THMs in drinking water, and this is really kind of what spurred almost this optimization of drinking water treatment, and that you couldn't just add an unlimited amount of chlorine anymore. You were limited by how many of these byproducts you could form, requiring you to provide better pre-treatment before disinfection. And starting in 1980s, predominantly in Europe, the rise of biological drinking water treatment started to take off and started to be seen in the literature. A lot of this work was actually more so around riverbank filtration and slow sand filtration. However, this was really kind of the first step in where we are today. And if we look at a Scopus review or a U of T library search for drinking water biological filtration, we see that in the decades before 2000, there was really relatively limited number of biological drinking water treatment papers published. And I think this is partially due to the limited number of papers published in general. But we start to see 2010, 2019, an exponential growth almost in the number of papers published in this area. So as we shift focus from chemical and physical treatment, we start to look at biological treatment and see the benefits that this really provides generally to the water industry. And I foresee this becoming a continued area of research because of the potential optimization and how little we actually know about these processes and how they work. So how do we implement biotreatment and biofiltration at drinking water treatment plants? In most utilities, we're going to consider rapid biofiltration using anthracite or GAC. I mentioned riverbank filtration earlier, and what this is really the use of a well located some distance from a water body. The water passes from a source water, such as a river or a lake, and it passes through the subsurface where it's collected by the well and pumped to a treatment facility. That contact with the subsurface environment acts as a biological filter where microorganisms can consume substrate and reduce the level of contaminant or substrate in that water sample. Another one that you might be familiar with is slow sand filtration. This is predominantly a small systems technology, generally due to the limited flow rate from these systems and the sheer size required for large-scale utilities. What this works by, you add your influent water at a low flow rate, a schmutz deck forms on the surface, which is a potentially thick layer of biological organisms and biological mass that then treats the water before being filtered through sand and gravel. When we're looking at typical municipal biofiltration, however, at places like the city of Toronto and other large cities or even smaller city treatment plants, we take water from a source, be it a river or a lake, and within that untreated source water, there are naturally occurring microorganisms represented by the green dots. As that water gets brought into the plant, there's some level of pre-treatment, typically coagulation, sedimentation. However, not all of those bacteria are removed or inactivated. So if we prevent a chlorine or ozone residual from entering our filter, we'll have some of those microorganisms attached to the surface of our filter media. They'll then start to attach, grow, form a biofilm, and form this biologically active layer. If we allow that to happen over a period of time and allow more and more biological activity to form on these filter media and promote the growth of biological processes, we start to form this EPS or extracellular polymeric substance layer, and we get a formation of a biofilm, which provides some biological treatment. So looking back to the 90s and 2000s, kind of when biological treatment first started to be evaluated in the North American context, a lot of the research was initially into how to operate these facilities. It was less about the specifics and more about how can we use these as utilities or as water providers. And really the focus for a lot of these studies was, how does backwash influence it? We've been using dual media or monomedia filters for decades, and we know how to operate it when they're used with chlorine, or we put chlorine through to prevent biological growth. But what's going to happen once they're biologically active? Are they going to form this concreted filter media that doesn't backwash? Are we going to run into operational issues? And that's what they wanted to overcome. Secondly, they also wanted to look at media type. So granular activated carbon has a much higher surface area per mass than either sand or anthracite. However, it's also much more expensive. A lot of facilities were able to get very good turbidity and particulate reduction using sand and anthracite, and that's what they had installed, but they wanted to see if there was a benefit to using GAC that justified the increase in cost. And finally, one of the biggest issues was water temperature. Biological filtration works really well in warm water climates. We talk to people in Texas and Florida all the time where water temperature ranges from 20 to 35 degrees C and they get great performance. And then they come up to Canada, they look at Lake Ontario, they see 4-degree water temperature in July, and they're like, okay, how does anything biological function? And that was one of the primary drivers for a lot of this early research, was to justify the use of this technology in a variety of locations and applications. Due to the science and the technology available at the time, the biological processes were also fairly limited. So they didn't have the analytical capabilities to do what we did now. So they kind of did the best that they could with the existing science. So biomass was often characterized using phospholipid analysis, and these phospholipid fatty acids are present in cell membranes. However, it doesn't provide any indication of whether those cells are alive or dead. A dead cell also contains phospholipids, and oftentimes these results didn't correlate very well to water quality because of some of the analytical and inherent limitations. Dissolved oxygen was often used as an indicator of cellular respiration. So just looking at how much oxygen was removed across a filter was commonly used as an indicator. And finally, the removal of organics was and still is really one of the drivers for biological evaluation. So assimilable organic carbon, how much of that bioavailable carbon is removed in a biological process. Ozone byproducts, simple carbon chains, oxalate, acetate, very simple carbon products, and that their removal was evaluated. And then TOC, what's just the bulk removal of organics. And these studies were great because they provided a lot of the fundamental information that drives our current understanding and really is the backbone of the existing research that we have. So just going through a couple of simple studies to demonstrate some of the science that was done. The first thing that everyone who doesn't have biofilters asks is, how long does it take to get biological filtration? How long am I going to have to wait before these biological processes are functioning and present? So on this figure, there are 4 different filters. There is 2 anthracite filters with or without chlorine in the backwash. And there are 2 GAC filters with or without chlorine in the backwash in a variety of temperatures. And we see that F2 and F4, the orange squares and the green dots, are the GAC filters, and they acclimate. They have a biological film and they have an active biomass relatively quickly. Within about 3 weeks, they see that there is an appreciable amount of biomass developed. When we compare that to the anthracite filters, the F1, or the lime green diamond shape, takes almost 12 to 14 weeks before it acclimates to the same level as the GAC filters. And that anthracite filter, the blue triangle, just never acclimates, and that's due to the exposure to chlorine. So we see that GAC as a filter media is generally better. And a lot of people look at this and say, okay, well, we need to go to GAC, we can't have chlorine, so we can or cannot achieve this at our plant. But when we start to take a look at the removal of organics, we see that even anthracite biofilters operated without exposure to chlorine can achieve similar levels of organics reduction. So in this particular figure, I believe it's oxalate reduction, but the anthracite and GAC filters show generally similar trends over the time period presented. And that really tells us that organics removal isn't biomass dependent as indicated by phospholipid concentration. So there's a lot of site-specific information that they developed and figured out that, okay, we may not need a lot of biological mass to get biological performance, and optimizing the performance with respect to eliminating chlorine is more important than filter media type. Another big issue was looking at backwash conditions. A lot of plants backwash from their clear well, so there's chlorine inherently present in their backwash water. And we can see on the left-hand side, the chlorinated backwash numbers are significantly lower than those with non-chlorinated backwash. If we just look at the biomass itself, we're about 10 times higher in biomass for non-chlorinated backwash. The other thing to note is when we have chlorinated backwash, we generally see an immediate dip in the amount of biomass. So that exposure to chlorine, some of that physical removal is actually impacting the amount of biomass on the filter. Ultimately, that lost biomass is recovered by the end of a filter run. And that's similar with non-chlorinated backwash as well. So what this tells us is that backwash really doesn't impact overall filter performance with respect to biological treatment. However, when we look at water quality, we do see similar trends. With chlorinated backwash, if we look at the removal of formaldehyde or other simple organics, immediately after backwash, we see a general reduction in performance. It may only be 10 or 15%, but that may be important for some facilities depending on what their DBP formation is. However, when we use non-chlorinated backwash on the right-hand side, we really don't see that same impact on performance following backwash. So there's a lot of this fundamental information that's been developed. GAC is better than anthracite typically. Non-chlorinated backwash is better than chlorinated backwash. And this is what's driven biological filtration design and operation for a number of years. And then in around the 2010s, roughly when I started my master's degree in 2012, there was this form of biofiltration renaissance. All of a sudden, a lot of new technology became available, and we started to reevaluate as an industry what we knew about biological filtration. And one of the main drivers for this initially was a rapid assay for adenosine triphosphate. If anyone remembers their Krebs cycle, ATP is kind of an energy carrier in cells, which is indicative in theory of how many cells are present and what they're able to degrade. And the hypothesis was that if we could measure ATP, we would then be able to make an inference about water quality based on the amount of biomass present. However, when we did this work, and this was published back in 2015 in Water Research, we generally saw that there were no correlations between water quality and ATP. We looked at organics reduction, DBP formation, potential removal, a variety of different organic fractions through LC-OCD, and didn't ever see a strong trend between the amount of biomass present and the inherent performance or removal of substrate. And the next thing that came from this was also maybe we can move from a passive biological process to an engineered biological process. So a lot of people looked at this and said, yeah, as engineers, why hadn't we thought about this? If we can look at carbon, nitrogen, phosphate ratios, we can make them carbon limited and really maximize the substrate uptake. And this was one of the formative papers for a lot of the research that I did as part of my MASC, but looking at engineered biofiltration and looking at nutrient and peroxide addition. And we did a study where we looked at a control biofilter just treating raw river water to see what the removal of DOC was. We compared that to a filter that had nutrient supplementation to ensure carbon limitations. We added hydrogen peroxide, we added inline alum, and we didn't see any statistical difference. These filters performed the same. It didn't matter if we added nutrients or other chemicals. We didn't get any benefit from those additions. We couldn't engineer biofiltration. However, the most important or the most effective treatment was to just go to GAC. It harvested, or it allowed the development of greater biomass, potentially more biological activity. And then there's also the concept of potential bioregeneration of absorption sites. The actual mechanism hasn't been well elucidated to this point, so I'm not going to comment on what the actual mechanism behind that is, but it just further confirmed that GAC provides better treatment than anthracite filters because of the physical media composition. And finally, and this is probably of interest to a lot of people because it's still a hot topic of discussion, there was a lot of thought that metagenomics would decode the biofilm and really give us some insight into treated water quality. So this is just a simple plot from data that we have and haven't published because we don't know what to do with it. But we can see that in general, through the depth of a filter from the top 5 centimeters to the bottom, there's generally less diversity. There's more alpha and beta proteobacteria. And these results are great, but this is generally what's seen throughout the industry. We can take samples from biofilters across North America and Europe, and we're generally going to see the same groups of bacteria present in all of them. And that's kind of been the challenge is, to this point, engineers have been unable to fully understand what this information means and utilize it to get a better understanding of the biological processes. So I think there's a lot of information here that's been underutilized and is definitely a path forward But up to this point, there hasn't been a good way to utilize this information in an effective way for operation or design of biofiltration systems. So what we learned from all of this, probably 20 to 30 years of research: chlorinated backwash should be avoided if possible. GAC media supports more biomass and gives better water quality. Ozone improves organics removal. If you can take large organics and break it down into smaller components with ozone or another oxidant, it often leads to better treatment performance. Biological processes are just better able to incorporate small, simple organics, and increasing temperature led to better performance. However, that's not really practical in most cases because it's hugely energy intensive to raise the temperature of water. So it's more of a site-by-site assessment and evaluation than it is an operational improvement. One of the biggest takeaways from this was that biofiltration provided robust treatment for a variety of parameters simultaneously. And that's probably the biggest selling point for biofiltration, is that it can remove a whole lot of different substrate at the same time. And that's a huge driver for a lot of plants that are looking to get wide-scale treatment of a variety of substrates. And finally, the biggest takeaway from a lot of this is that biomass isn't indicative of performance. We don't know what the treated water quality is going to be based on the amount of biomass alone. So that's really what drove a lot of my research and will really lead to where we are now. So looking at biofiltration in 2020, generally we have a pretty good understanding of how things work. As an industry, we know what it can remove. We have a general understanding of what the percent removals might be in any given situation. We know the substrates that it can and can't and the conditions required for each of those removal pathways. But really what we've moved on to is trying to better understand biofilter function and biological function specifically. So looking at how to characterize and quantify the biodegradation capacity in addition to biomass density, or as a replacement to biomass density. So we've done 8 years of biofiltration piloting at a variety of plants. We started with the Peterborough water treatment plant in 2012 and added ozone a few years after that. We have 8 biofiltration pilot filters under a variety of different media and backwash regimes. Each of the pilot filters is backwashed with its own effluent, or can be replaced with chlorinated effluent, so we can test upwards of 1,000 different combinations of ozone, non-ozone, filter media type backwash conditions. It's a really great treatment facility to be able to test some of these systems. There was a pilot at the Toronto RC Harris plant. We have one at York Region in Georgina. And this plant is particularly unique because it actually has ultrafiltration membranes prior to biofiltration. So their biofilters act more as biological contactors than they do biological filters. Peel Region, Lakeview Water Treatment Plant. A note to anyone doing biological testing: this plant's beautiful and has huge windows, but because it has huge windows, it also has huge algae problems. So you can see that all of the components are wrapped in black fabric to prevent any algae growth throughout the system. It's a real pain, but something to keep note of in your lab is if you want to do biological work, avoid exposure to UV light. Elgin and then Barry, and similar concept, cyclical operation at this facility following ultrafiltration membranes. And on the right-hand side of that picture is actually a UF pilot system that we've connected our biofiltration pilot to, so we can simultaneously optimize UF and biological treatment performance. So just kind of summarizing some of the work we did, this was at the Georgina pilot plant. We compared continuous and cyclical operation at 2 different empty bed contact times, so 2 different flow rates. And we see that generally cyclical operation is similar or better. And what this tells us is that cyclical operation changes the oxidation regime from an oxygen-rich to an oxygen-deprived environment by shutting down the plant for 12 to 15 hours a day. And the concern from the operations team at the facility was that shutting down the filter would actually kill all of the biomass. And what we found was that it actually did the opposite, and it actually improved biological performance by selectively promoting bacteria that can survive trying environments. And this was relatively new to the water industry. Any of you doing wastewater research are probably laughing at this and saying, well, yeah, we've known that for probably 50 or 60 years, that going between anoxic and aerobic environments can promote biological activity and provide function that we don't otherwise have. But this was kind of one of the first times in drinking water that we saw similar results. Looking at the Peterborough pilot plant for a variety of different organics, We looked at different media types, either anthracite or GAC. We looked at settled water compared to ozonated settled water, and generally found that using ozone and GAC was far superior for the removal of all of our organics, and longer empty bed contact times or longer retention times were superior as well. So this kind of all summarized and confirmed the results that had been published previously and ensured that the results we were getting were kind of valid. But what we wanted to do was really take this to the next step and see not only what can it do, but why can we expect this performance? And one of the things that I started with in my PhD was looking at enzyme activity. And enzyme activity is really just indicative of a rate of degradation for specific substrates. In this case, the assay we used uses a fluorescent 4-methylumbelliferone substrate backbone on the left-hand side. We can then replace that hydroxyl group with different substrates for degradation, such as phosphate or acetate, and those different functional groups are then indicative of various enzyme activity. In this case, 4-MUB phosphate is indicative of phosphatase activity, and 4-methylumbelliferyl acetate is indicative of esterase activity. And the hypothesis we went with was that enzyme activity was an indicator of biological treatment performance and could be related to organic or other substrate degradation. So when we first quantified this, we looked at anthracite, GAC at various contact times, and we saw what we would expect. From going from anthracite to GAC, we saw a general increase in activity. And that makes sense. There's more biomass on GAC, and this confirms that we're also getting more biological function in terms of esterase activity. Looking at the blue bars for GAC 15 minutes and GAC 30, we see a decrease. And again, this made sense to us. When you have a 15-minute empty bed contact time, that means you're providing twice as much substrate compared to a 30-minute empty bed contact time. And that's giving us the ability to then promote more biological growth because there's twice as much substrate for consumption. When we look at ozone, we see a very similar trend. It's just elevated relative to settled water. And this is attributed to more easily degradable carbon compounds being present following oxidation using ozone. Taking that information was great, but we wanted to see if we could then correlate it to water quality. Up to this point, it was very similar to ATP in that we could figure out the relative quantity and compare processes, but we still couldn't just directly take this information and then correlate it to water quality. The correlations just weren't there. So what we did is we developed a term called effective activity. And if you notice, enzyme activity has units of micromoles per minute. Empty bed contact time is per minute. So we multiplied these out to get a biological degradation potential in micromoles. And this unit is particularly nice because it incorporates filter design, operation, and substrate availability into one parameter. Having anthracite or GAC media will be indicated in your enzyme activity. Your flow rate will be reflected in your enzyme activity. If you have ozonation, that's reflected in your enzyme activity. So all of these terms come together and are really well represented with this one parameter. And effective activity was then compared to DOC, THM, HAA removal, as well as phosphate removal for phosphatase activity. And when we looked at these, we see pretty good relationships to organics removal. If we look at ozonated water, we see a fairly strong relationship as well as settled water. And this is over a variety of different treatment types, different filter compositions, different water temperatures. So to be able to look at this over a period of a year and see that we're getting relatively strong increasing relationships is really telling for us and promising that enzyme activity is a good way to move forward in evaluating biofilter function. Similarly, at Georgina, we saw that when we use cyclical operation, the red lines, we get moderately elevated enzyme activity relative to the continuously operated filters. And this is even more telling that the enzyme activity is similar between these filters, despite the fact that the cyclical filters had roughly half the biomass that the continuously operated filters had. So the bacteria in the cyclical filters were preferentially able to produce the activity and the function when compared to the continuous filters. And when we look at the effective activity, again, for each filter, we see strong linear relationships. And this was later then also considered more holistically, and we found strong relationships in subsequent studies with cyclical and continuous operation, regardless of flow rate. Looking at phosphatase activity, we also see strong linear relationships between those. So the removal of phosphate and effective phosphatase activity were strongly correlated. And this isn't to say that either of these are causal relationships. We're not entirely sure what the mechanism is, but enzyme activity seems to be a good surrogate for treatment performance. And this kind of brings us to what we currently know, or kind of the current state of the industry with respect to biofilter function. And that enzyme activity provides us a way to quantify function and really, for the first time, understand some of the mechanisms that are occurring within the biofilm in a way that engineers can start to interpret that data. And it effectively and nicely accounts for filter operation and design. So instead of having to concern yourself with all of these different configurations and do all sorts of different regression analyses, we're hoping to move that towards a simple parameter that can be used regardless at a facility. If we combine enzyme activity with filter operation, specifically contact time, it may be indicative of degradation potential. We found that esterase was related to carbon production, phosphatase was related to phosphorus reduction, and other research has identified specific pathways for other substrates of interest. And this is of particular importance for things like nitrification or manganese or iron reduction. Some of these pathways are very well understood. Things like manganese and iron reduction and oxidation can be traced back to individual organisms or individual enzymatic pathways that facilitate those reactions. If we were concerned about those particular pathways or those particular substrates in a biological system, we could specifically monitor for those enzymes directly. And I think that's really nice that we can start to do substrate-specific monitoring for those pathways, and the work will continue to look into identifying those for things like disinfection byproduct precursor reduction. Finally, the limitation of all of this is that biological processes are generally still site-specific. So we were able to develop these relationships for individual water treatment plants, but it doesn't seem like these are necessarily able to be replicated at other facilities without some sort of interpretation or resampling. And I think a lot of this comes down to just variation in substrate. Esterase is a very general enzyme. However, the organic composition of each facility will change significantly and will likely have to drive specific relationships being developed. So where we're going, and I think this is where everyone wants to get to see kind of what the next steps are, what I feel might be some of the next steps. And I think the biggest thing from an industry perspective is the development of reliable control mechanisms and an ability for operators to optimize these biological processes in their plant. We found that cyclical operation, shutting the filters down for a period of time each day, may provide some benefit. It may increase function relative to biomass, but we still don't know how much shutdown. How long does it need to be shut down for? Is it related to the amount of dissolved oxygen available? What is it within that process that promotes function that we're looking for? And that's really the next big step from an implementation standpoint, is how do we get to that point of being able to tell an operator how to optimize their process? What do we need to monitor? What are the keys to operation in a full-scale facility? Secondly, understanding the relationship between biological function and biodegradation potential. And this is really kind of the big step. Is up to this point, everything we've done has just kind of been a surrogate. We've been able to measure esterase activity. We've been able to relate it to carbon degradation, but we don't know whether or not esterase enzymes are actually responsible for the degradation of the organics that we're seeing, or if they're just well correlated because they're a general enzyme indicative of general function. So there was actually a presentation at the CAWQ virtual conference last week that I found really interesting, where researchers in 2017 developed a nanofiltration membrane bioreactor for wastewater treatment, added lactase enzymes to this bioreactor, and then looked at the degradation of a variety of trace organic compounds. And they were able to find that based on pKa and log D, they were able to find correlations between the effective treatment and the degradation by lactase directly. So not only were they able to attribute the degradation or the biological removal of these compounds to lactase directly by exposing them to a pure enzyme, they were also able to figure out the chemical properties that most likely led to them being degradable based on physical chemical properties. And looking at this work is really a precursor to some of the work that we're going to be doing, looking at drinking water contaminants such as DBPs or dissolved organic carbon and exposing them directly to various enzymes to see what the degradation potential is. And all of this is in an effort to get to a monitoring parameter that tells us the most regarding specific biological pathways that can be easily monitored. And finally, I think this is kind of like my 25 to 30 year project is genetically modified water. And I think this is an interesting topic potentially because a lot is known about the degradation of specific substrates. So there are some bacteria that provide function that no other bacteria provide. However, the bacteria that we might be looking for, perhaps nitrifiers or manganese reducing bacteria, may not be naturally present in a source water, and we'd like to harvest that function. So I think the potential is there to extract the DNA from bacteria that we're interested in or have function and implement that into bacteria that are naturally present. A lot of the questions I usually get around biofiltration are, can we not just seed biofilters with bacteria that degrade what we're looking for? And the answer is, there have been numerous studies that have tried it and it never lasts. Unless those bacteria are naturally present in a source water, it's unlikely that they're going to acclimate on the biofilter and have a sustainable population in a week's time after you stop dosing those bacteria. If we could then impart or somehow implant the function we're looking for into naturally occurring bacteria, the proteobacteria that are commonly found on biofilters, then there's a potential to move forward in targeting the specific treatment we're looking for without having to do supplementation or find other ways to degrade it. So I think it's really interesting that the potential might be there, and it's something that things like CRISPR might make a reality in the decades to come. Although there's going to be a lot of questions around what genetically modified water means and how you would get utilities to be able to effectively market and get consumers to buy into that. So with that, I need to thank all of the NSERC chair partners, everyone that we've worked with over the last 10 years.